Hi, I'm Jeremy. I'm with Jennifer here and Avery. You might recognize Avery from Answers News. And if you don't recognize her from Answers News, that runs Mondays and Thursdays on the Answers in Genesis Facebook page. So I encourage you to check that out. She appears there from time to time. Anyway, you might notice, if you don't, it's because they're tiny on the small screen. We have new name tags. Mine says Jeremy, yours says Jennifer, and Avery says Avery. So isn't that wonderful? So if you come to the Creation Museum, you will know who you're talking to because we all have name tags now. So that's really cool and helps make it more personal. Anyway, we are here for something exciting.
Yes.
Jennifer, what do you do here at the Creation Museum, and what is all this?
I work on preparing educational programs for students, and so next Friday we have a sold-out event here called Explore Botany, and this is just a small sampling of what's going to be happening next Friday, and we're actually going to work through a couple labs today to show you what's going to be going on.
And we also have Explore camps this summer for June and July, and we'll be talking more about that, but let's get into the good stuff right now. So let's go ahead and start with the experiments.
Okay, fantastic. So we're going to begin with a flower dissection and talk about some flower anatomy. So it's very important whenever you do a lab to always have safety first. So Avery, we are first going to have to put on our safety goggles today.
All right. Very fashionable.
Yes. And we need to put on safety gloves.
Thank you. Also very fashionable.
Yes.
All right. So what kind of flowers do we have?
We
Look
have
like
lilies today. Okay. These are always difficult to get on. There we go.
There we go.
Okay, so yes, we're going to be dissecting lilies today. And so it's very important whenever you're using dissection equipment and scalpels and things like that, that you are safe. And so that's why we put on our goggles today, just in case there was to be an accident. So we are dissecting lilies today, and these are lilies, all right, and you can see how beautiful they are. So Avery, go ahead and just break off a lily right there, and we're going to lay that on our dissection pan.
Alrighty.
Okay. Now, the first thing you want to do is look at the different parts of the flower that we can visibly see, all right? So the first thing you can see is its petals, all right? And the lily does have 3 true petals that you see here on the top, okay? And then it does have 3 separate petals, or what we call sepals, underneath, all right? So you want to go ahead. The first thing we need to do so we can get to the internal structure is you're going to carefully peel off your petals as carefully as you can near the stem and your sepals.
They are covered in pollen.
Yes.
Oops. There we go. All right. Doesn't
Okay.
look quite as pretty now.
No, it doesn't, but we can see the interior of the plant better now. All right, so now very visible to us are 2 very important things, and one is the male parts of the plant, and one is the female parts of the plant, which are very important. All right, so the first one is we'll take a look at is the male parts of the plant. All right, and so the male parts of the plant is comprised of 2 parts, and that's the filament and the anther. So you can see here this long portion right here, this is called your filament, okay? And then this thing here that's, slowly turning into pollen and about to fall off is your anther, okay? And that's the male part of the plant. It is together, those 2 parts are called the stigma, all right? I'm sorry, this is the stigma. These 2 parts are called the stamen, all right? And a good way to remember that is that stamen, stamen. So it helps you remember that that's the male part of the plant,
All right.
okay? Then if you look at the interior, this is your stigma right here, and we're going to be looking at the female parts of the plant, all right? So what we have on top is our style, all right? And that's the sticky little entry point that you see here. Then we have this long part called the style, and it actually joins into our ovary, which if you carefully move your anther to the side, you will be able to see your ovary down here near the bottom of the stem. Yes, okay.
Cool.
Now, lilies would be considered a complete flower because the male parts and the female parts are in one flower together. So it's very easy for them to pollinate. All right, so you can see how closely the pollen granules are here to the actual stigma where the pollen, has to enter, all right, to fertilize the ovary down here. And what's really interesting is that every individual flower will only allow the pollen granules down the stigma when it recognizes that it's a specific plant.
Wow, that's
Okay?
really cool.
It is.
Wow.
Because if pollen lands on the stigma that's from another flowering plant,
Right.
it's not going to allow those pollen granules down to the ovary to fertilize, all right,
I
to
would think that happens
reproduce.
pretty often with wind and
Because,
insects and whatnot.
right, because you know what it says in Genesis? That plants
It's according to their
reproduce
kind.
according to their kind. So it makes sense that a plant would recognize, and it actually secretes a chemical signature on the stigma that will either permit or not permit pollen to go down the stigma
Wow.
and fertilize the ovary. So it's pretty cool.
That is really
So the
neat.
first thing we do is we're going to peel off our anthers, right? So you can peel off your anthers and your filaments or your stamens, all right? And this particular plant, you can see, has how many stamens, Avery?
6, I think. I knocked 2 off, so I'm 2 off at 6.
All right, so now we're just left. We've taken off the male parts of the plant. Now we're just left at the female parts of the plant. All right, and so this is where we want to kind of cut into it and see the actual ovules that are inside the ovary. All right,
Right.
so very carefully, you want to take your scalpel, all right, and we want to sever it from right here at the base of the ovary and go ahead and just take off that whole female piece from the stem.
Okay.
Okay?
Maybe got a scalpel in here.
So I get woozy at the sight of blood. Will I get woozy as you kind of dissect this plant?
No, there are no plants, Jared. I mean, plants do not have blood because they're not what we say nephesh, right? And so they are alive, but they don't have the breath of life. They don't do not
that's
have
really-
blood.
thank
So
It's right here.
yes, so go ahead and cut that
All
right
right.
there. All right, so now we've removed the stem, and now we can actually see. Right. So now you want to remove the stigma from the ovary
Okay,
itself. So
so
go ahead and
right
just,
here?
yep, just cut right there.
That's easier when you
Okay.
use the sharp side of the knife.
It is, yes, very important. All right. So now to actually see the individual ovules inside, you want to do like a longitudinal section. So you're going to, cross section, we'd be cutting the short way, but we want to look at it long ways. So go ahead and just do a careful cut, but don't cut, try not to cut just one single cut, let's say. You don't want to make
Okay.
multiple cuts,
Oh boy.
because we don't
All
want to
right.
damage the inside.
Careful, don't damage it.
Okay.
Okay, so go ahead, take your forceps, all right, and we're going to carefully open that up.
Oh, maybe I didn't cut deep enough.
Maybe you should have been a plant doctor, Avery.
Yes. Oh, I may need to try my
Yeah,
incision again.
you might need to.
All right, let's try this again.
You don't want to hear a doctor
I
say
think I'm
that.
being too careful. Okay, there. Let's try that. All right. So plants are pretty complex. You got a lot
They
of
are.
parts.
They're pretty complex.
cool.
Yeah.
Apparently tough to get into. Okay. What am I doing wrong?
Are you
Did
sure?
I just not cut? Do I need to cut like all
Are
the way
you-
through?
Yes, you need to cut all the way.
Oh,
yeah,
well,
yeah. One single cut. You're going to cut. You're not cutting deep enough. That's what
Okay,
it is.
okay.
Right.
So cut right through to the
Yeah. Go
tray.
ahead. Let's go
Okay.
ahead so we can open it up.
Alrighty. There we go.
Yay!
Yay me!
Okay, great. So now what we want to look at, so remember this is the ovary, right? But inside there's going to be
Okay.
ovules or eggs that get fertilized by the pollen, all right, that comes down through the style, all right? Here's your style here, okay? Down into
Okay.
the ovary. So go ahead and get your magnifying glass, because sometimes they're really, I mean, they're extremely small. So you're going to have to look very closely, but you should see down here, let me see if I can find a little pointer here. There we go. Down here, where you see this darker green area right there, if you look closely, you might be able to see the little ovules. And they look like little tiny white eggs that you see.
Jennifer, while Avery is looking for that, we do have a really good question from Carrie. She asked, is the camp for students going into the 4th grade, or do they have to already completed the 4th grade? So, in the summer, if they're going to 4th grade next school year, is
Right.
that fine?
That is a very good question, but they do have to have already finished 4th grade.
Okay.
to be permitted to enroll in camp.
Thank you. That's
But
helpful.
they can definitely come next year,
Sure.
and we'd love to have them, for sure.
That
So,
is so
what do you
cool.
think, Avery? Can you see the little
think so.
ovules? And there's usually multiple, depends on the flower. You should be able to see at least 6 visible in here, right here.
That is so cool. Yeah, I see at least one.
Now, in incomplete flowers who do not have the male and female parts, right next within the same flower, then they have to rely on insects or the wind or something else to pull those pollen granules to that flower. But the same principle applies. If it doesn't recognize the pollen from its species, it's not going to permit the pollen to go down the stigma.
That is really
All right.
cool.
So that's really neat how flowers are. Okay?
Now, if
All
you have
right.
any questions, comment below, and we'll answer them as we're doing this experiment here.
So what about the leaves of the plant?
Okay, well, we'll take a look at leaves microscopically. So if you want to move over to the microscope.
Take off my gloves?
Yes, you could take off your gloves. It'd be okay. As long as
Didn't
you're
wanna
not,
get
as long as
pollen
you're
on
not allergic to clear nail polish.
Nope.
Okay, then you're good to go. Don't want you to have an allergic reaction.
That wouldn't be good. No, I'm good.
All right, so what's really fascinating about plants, all right, is that they each have what we call individual fingerprints, all right? And they have these stoma patterns that are unique to each species in a plant. And what's really interesting is that paleontologists have been studying leaf stoma in the fossil record, and what they've discovered is that the leaf stoma we see in the fossil record exactly matches those plants that are still alive
That's
today.
really cool.
The stoma hasn't changed in millions of years, and the evolutionists can't explain that because we would think there would be some changes, but the
Right, yeah.
patterns almost exactly match what we see in those plants that
Wow.
are living today, which would make sense to us,
Yeah.
because God created after their kind, and we would expect stoma
within a
to,
few
yeah,
thousand years,
right?
yeah.
We would expect to see the patterns the same. So you're going to look at 2 different stoma patterns today. But before we do that, I just kind of like to explain to you what you're actually looking at.
right.
So if we were to take a leaf and take a cross section, that means we're going to look at it from the side. And of course, they're extremely thin. You'd have to do this microscopically. But plants basically have 3 general layers. The middle layer, the mesophyll, is broken into 2 parts, but they do have the upper epidermis, the lower epidermis, and then these mesophyll layers that are broken into 2 in the middle. Okay? What's really fascinating is the upper layer of the leaf, the epidermis of the leaf, is almost transparent, which would make sense because sunlight needs to
Right.
get through that into the second layer where most of the the chloroplasts are, and they need chloroplasts to absorb the sun, right? So they can produce energy, right, for photosynthesis. So down here, though, on the lower epidermis, or what we would think of as the underside of the leaf, is where the stoma are located.
Okay.
All right? So if you were going to take a stoma and kind of look at one, all right, this is what you would see right here. And they almost look like little tiny mouths that have the ability to open and close depending on what time of day it is. All right?
Okay.
So when it opens, it does permit gas exchange, right? So plants, do you remember what they take in?
They take in carbon dioxide.
Right, and they release oxygen. So carbon dioxide and oxygen are going to be flowing in and out of these stoma while they're open, all right, in addition to water, all right? Well, that can be an issue because plants need to conserve their water,
Right, yeah.
Because, they require a lot of water, and they have a vascular system. So think about this now, all right, if they're releasing water, all right, but they do have to engage in photosynthesis, and they have to go through gas exchange, what time of day do you think the stoma might be open?
I think during the cooler hours, like in the morning or the evening, when there's less evaporation of the water, probably.
Right, well, it would be during the day because that's when the sun's out and it needs to get the sunlight. And what's really fascinating is in the lower epidermis that we have here, this underside, there are no chloroplasts present except on the outsides of the guard cells, all right? And that kind of helps give it the energy to go through this open-close motion and allow the gas exchange and water vapor to exchange. But at night, it does close up, right?
Okay.
There's no sun, okay? It conserves its water, all right? But what about desert plants? What do you think they do?
That's a good question.
Because it's so dry in the desert,
yeah.
right? Do you think they'd be open during the day or possibly at night?
Probably at night. to keep the
Yes,
water.
right. It would be at night. And what's really fascinating about desert plants is they know and they have the ability to conserve their photosynthesis or their sun energy they're absorbing during the day and then
Wow.
open the stoma at night when it's cooler so they
Like
don't
delay
lose
the
their
process
right
until
so they
night.
don't lose the
Wow.
water vapor in the plant. It prevents them from drying out. So it's
That's
really
really cool.
fascinating. All right. So you're going to take a look at these because what makes these special is that the pattern on each species of plant is different, and we can look at the stoma patterns and know what plant it came from because it makes like an individual fingerprint. All right. So the first way you're going to do this is to just break off a plant from each, a leaf from each of the 2 plants here. And these are both just your typical house plants. This is a great lab kids can do at home. If you have a microscope and some plants and some clear nail polish, that's pretty much all you need.
And these are healthy plants, so if you're concerned regarding the plant's well-being, they will be able to be without one leaf.
No
Yes.
plants were harmed in the making of this video.
No, right. No plants were harmed at all. All right, so you're going to take the clear nail polish, and you only need to do like a very, very small square of clear nail polish on the underside, right? Because the stoma are located on the underside, not the upper
Which
epidermis.
feels different than the
Right, because there's a waxy, waterproof type substance that's on the top of leaves so that the water. Mhm.
All right, so just pick a spot. And
Just
put
pick a
little
spot
square?
and do just a little tiny square, fairly thick, but not so thick that it takes a long time to dry.
Okay. Like
That's
that?
plenty. Mhm.
Good.
Mhm.
All right.
Now we need that to dry, so kind of just like blow on it, but don't blow your leaves off the tray. We kind of want it to dry. Yeah, so just
See if
kind
this goes better than using the scalpel.
So just blow on that. This can take a couple minutes depending on what brand of clear nail polish you have. I don't think this one's a quick drying. So.
And you wouldn't want to put nail polish on the actual living leaves connected to the
Right.
plant.
You wouldn't want to do that. No.
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 Wouldn't be so good.
I mean, it might make it beautiful for maybe a couple days, but then I think the leaves would
Right.
die.
Yes, it would, because it can't
Yeah.
breathe anymore. It blocks the pores, because a stoma is kind of like a pore, that allows water and gases to go in and out of the plant.
Got lots of practice.
Now, in
doing
aquatic plants, where do you think the stoma would be located? Do you want to take a guess? Because it is different in aquatic plants compared to land plants.
That's a good question, because most of them are underwater. So.
Right. And the stoma reacts, it does have chloroplast, so it does react to a certain amount of energy from the sun.
Right.
So in aquatic plants, the stomas are located on the upper epidermis of the
Okay.
leaf. Mhm.
Wow.
Yeah.
I was just about to guess that.
Or-
Does that give them having it on the top instead of on the bottom? Just they can have more chloroplasts on top?
Right,
or-
well, all the chloroplasts are on the outside of the stoma, and so they're located on the top of the plant, so they can get that sun energy since they're underwater like that. Can I put your finger on it and see if it's drying?
Still
Not
sticking to
quite,
my
yeah,
finger.
okay, let's just keep blowing on it.
So while that is drying, it's like watching paint dry, however, it is nail polish. Anyway, so we are experiencing some of what you could experience, students grades 4 through 12, what you could experience this summer, either June or July. This is the botany, so botany is specifically for the July camp. However, let me talk to you about the different topics that will be covered. In June, you're going to be discovering genetics, geology, forensics, astronomy, and zoology, and you're going to be doing all different types of things similar to what you're doing here, just, for those different topics. And then July, not only do you have botany that we're discovering right here, right now, you could be experiencing this, but also forensics, human anatomy, astronomy, and zoology as well. So some of the same topics, but there's a little bit of difference depending on the interest of the student itself. So, but here's what you need to do. By May 16th, you need to have registered for the June camp because May 16th is the deadline. We need to be able to get scheduled for all the food and everything that's going to be happening. So be sure if you're interested in coming in June to have registered. That's coming up really soon. And then May 27th for the July camp. So just be sure to register by then. Talk about it with your families and let's get this done. So how's it going?
It's almost ready, I think. So Avery, you can go ahead and turn your microscope on. We can get your microscope ready. It's right here in the front. Yep.
All right.
So whenever you're using a microscope, it's very important to always make sure you start at the lowest objective. All right. And for most microscopes, that is objective 4. Basically, what that means is you multiply whatever the number is on the objective, and you usually see a little number here on the side of the objective. You multiply that times the magnification on your eyepiece. And so we see here there is a 10. So it's 4 times 10, so you're seeing
40.
40 times larger. Right, very good. So you always want to start at the 4 because you get a more big picture view of what you're looking at
Okay.
microscopically before you begin to focus. All right, so I think it's about ready. So you're going to get the tape out of your kit. It's very important that you use clear Scotch tape for this. Scotch tape or any kind of generic brand is fine. You don't want the one that has that's kind of frosted. It's
Oh,
better,
right,
right?
yeah.
It's better
Clear,
to
clear.
use a completely clear tape.
Okay.
So go ahead, take a small piece, and you're going to lay it over it's almost like a cast of the fingerprint or the stoma that you're taking here. You're going to lay your tape over it and rub the tape over the place where you put the clear nail polish, and then carefully go ahead and get your microscope slides out of the box. All right, and so when you peel that piece of tape off, you just want to lay it right on the microscope slide.
All right. Let's peel the tape off. So we're not actually looking at the leaves themselves. We're going to look
We're going to look at
cast,
we're
don't you?
looking at the
Okay.
the
And put it
stomata.
sticky side down?
Sticky side down. You can lay it right on there. That way it doesn't go anywhere. So what we're looking at is the stomata. All right, and looking to see how prevalent it is. Are they open or closed? All right, those are things you want to look for.
Okay.
And a great experiment to do at home, kids, is to keep one plant in a closet and one plant outside for a day, and then look at their stomata the next day, because I'm sure you can guess if you keep a plant in the closet all day, what happens to their stoma?
They close because
They
think
close, right? So you can get a good idea of what the difference is if you run that experiment at home.
Cool.
So, all right, so go ahead, pull back your stage clip. That's your stage clip, and there's a little handle right. Yep, pull that back. And we're going to lay our slide. You always put your slides on your stage. It's kind of like dancing. That's where all the, activity happens. It's the same thing with a microscope.
Like that?
Close your
All right.
stage clip. And it's always a good idea to get what you're trying to look at under the light source before you look at it through the eyepiece. And then over here, these are a mechanical stage. So over here you see 2 knobs. Do you see those?
Yeah.
One knob will allow you to move right and left, and one will allow you to move up and back.
All right.
So you want to go ahead and get that right over the light source.
There we go.
Okay?
That's about right.
So go ahead, take a look through the eyepiece.
All right.
Most likely it's going to look blurry. And so these are your focusing knobs right here. The large knobs are called your course adjustment knobs. They allow you to focus what you're looking at. So just move that up and down. Put some force on it, Avery. Got to turn it a little harder than that.
Okay. Oh, there we
There
go.
you go.
It's still just 2 white dots.
If you need me to help you, I
Something
can.
wrong. Yes.
Okay, let me see. Here, let me take a look real quick. Let me see.
We'll just pretend I'm in
Yeah,
4th grade.
We don't even have white dots here. Hold on. Okay, hold on.
But were they cool white dots?
They were cool. They were cool. Very cool.
Okay, these
I'm
are
still seeing them.
teeny. Okay. Now, take a look at that. They're extremely small right now. Okay. Because remember, stoma are like almost microscopic. You can't really see them unless we look at them under the microscope. All right. Remember, they're going to be white and clear. Okay. So what you need to do now is go to higher magnification because we really can't see it that far away, can we?
Right. Yeah. It's just
So
little dots.
let's go to 100 times. So that's your 10 eyepiece times 10. So it'd be
Okay.
100 times. That's this one right here. And
Okay.
just rotate. So just rotate that down.
All
All right. Take a look again. And then we probably have to focus a little
Yeah.
again. Okay. You can probably just use your fine adjustment knob,
Okay.
which is right here. You can try that first. You may not need to use the large knob to get it focused. If you need help, just let me know.
Sort of. See? Oh wow. All right, it's coming in now. There
Oh,
we
good.
go.
Okay, good. All right, so now we just want to see it really close, so we need to just go up one more magnification because you really need to be on 400 times to see them clearly.
Okay.
And so that's your 40 objective. So just go ahead and rotate that one more time.
All
And
right.
focus one more time. And let's see.
The kids at the camp are gonna love this. This is really
I know, this
cool.
is always one of their favorite labs is looking and seeing the difference between the leaves and how each pattern is distinctly different.
So what are you seeing, Avery?
I see my eyelashes at the moment.
Using a microscope
Which are
sometimes
cool.
takes a little practice. You know, I think, you know what, Jeremy? I think Avery needs to attend the Explore microscopes that we're having in the fall. Because we're going to have a whole day devoted to learning how to use a microscope with Dr. Menton, and I think Avery needs to come to that.
I think I need to.
There you go, Avery. We can schedule you in.
Yes.
Okay, I'm going to let you
Okay.
do this because I just see my eyelashes.
Oh, you have nothing there, Avery. Literally nothing.
Well, it's the microscope error, clearly.
Always
Right.
microscope error, right?
Always, always.
Okay. Now, because it's a leaf and it has different layers, things kind of go, you can go in and out of focus. That's why sometimes people get confused with that. But go ahead and take a look at this. Okay, and then we'll look at the print from your other example here, and you should see a
Alright.
distinct difference.
You're much better at this than me. That's why you lead the workshop. Wow, that is really cool. Wow.
You're not talking about your eyelashes?
No, I'm actually seeing something.
Yes,
else.
she is looking at the right thing now.
I can see all the mites that live on
All
my
right, so go ahead and take another piece of tape. You're going to
Really cool.
peel it off this leaf, and let's
Okay.
take a look at one more, because I'd just like you to see the difference. That's the important part of this particular lab. And of course, when the kids come, they'll be looking at more than just two. Spinach has a really beautiful pattern, so we'll be looking
cool.
at those as well. Spinach is kind of interesting, especially if you take it out of the fridge and look at it. Look at their stoma. It's interesting to see how many are open and closed.
Is that the example on the screen of what she's looking at?
Yes. So if you were to look at the two stoma, all right, you can see when it's open and closed, when that pore is actually open. So when it's open, right, you're going to see like this opening, all right, and then when it's closed, it's going to look sealed. It's almost like a little mouth that has its lips closed is what it looks like when it's sealed.
Cool.
Most of those were open because I had the plant light on them all night in my office, so they weren't in
Okay,
the
so
dark,
they-
so they're
all right.
right, their stoma's open. All right, so yeah, just go ahead, pull the stage clip back. Oh, wait, first, always go back down to your lowest objective.
All right.
We don't want to break a slide, so there you go.
Yeah, I prefer not to break anything too badly anyway.
So not only are we learning about botany, we're also learning how to use a microscope.
Yes.
And you can experience this summer at the Explore Camp. So this is one of the things that you'll be experiencing in the July Explore Camp.
Yes, so go ahead. You're going to focus that again. And what's really great is when they come, they leave with a whole packet of resources. So this is just an example of the Explore Botany resources the kids will be working on when they're here next Friday and at the Explore Camp this summer. And they get this packet of resources for every single topic that they get to participate in.
So not only do you get the experience, but you get a lot of resources to take home.
To take home, right.
Yeah.
Are you getting better now?
A little bit, I
Oh,
think.
good. You'll be an expert by the end of the workshop.
And with the workshop and the summer camps, we will have people here to help each student to be able to,
it
be able
won't
to
be me.
learn how to use these. And it will not be Avery.
Yes. All of our trap
Yeah,
is
you're
staying. Yeah. All of our our staff goes through training the day before at Explore Day so that they're ready to help the kids when they come. You have nothing here, Avery.
I know, I don't know what I keep doing wrong. I'm pretty sure this isn't what it's supposed to look like.
Now, what you'll notice is they're
like snow.
far more prevalent in this one. Let me see if I can get this a little better for you. Well, I think it's good enough for you to get the idea. Go
All
ahead.
right.
Okay.
Much better. You're
Yes.
a genius.
But do you see how they're far more prevalent than they were on the
Yeah,
pothos
yeah.
plant? This is a pothos plant right here. Yeah, it's far more prevalent on this little ivy bush than it is on
Wow.
on the pothos plant. Yeah. So that's just a small sampling of just what the pattern looks like on just
really
two.
cool. Yeah.
So yeah, if you come to
Wow.
camp, you get to look at more than that. So yeah, it'll be a lot of fun.
Come.
Yes, it's going to be a lot of fun. Definitely.
So now the question is, Jennifer, is Avery good enough to be able to get our certificate of completion?
certificate.
I'm a little hesitant to give this to her. But every child who comes to an Explore Day or Explore Camp does get a certificate of completion for that topic. So we are going to present Avery with her Explore Botany certificate, even though I think she needs to come back.
Yeah, I think.
For Explore microscopes.
Maybe just
Yes.
the microscope.
Yes,
I
I
think
think
I got the plant
so.
part okay.
I think-
Well,
yeah, I
other than
think you did.
Maybe, is there a scalpel class?
There is. In fact, we're dissecting in the human anatomy
Oh, okay.
in the July camp. We're dissecting sheep parts.
Oh,
So
wow.
you
Cool.
could come in and get firsthand experience with the scalpel.
Awesome. That
From
sounds good.
from Dr. Tommy Mitchell, our medical
he's a
doctor on
medical
staff.
doctor, yeah.
Right. So
Awesome.
yeah.
And
be a
you
great
can
opportunity.
come back multiple times. So if the student really wants to get very proficient at something, you can come back multiple times.
Yes.
You get a certificate of completion. You could frame it. You could get like a clear plastic tablecloth, put it underneath, so every time your family sits down to eat, you could say, look, I got a certificate of completion at the Explore camps and
Yes.
Explore workshops. So there's multiple ways that you could utilize this
There is.
and your
Yes.
other resources, along with
Absolutely.
the experiments, learning lots of great
Yeah.
things about science, God's creation, and how that science is consistent with God's word. It is not something that we find to be incompatible with each other.
Yeah,
Right,
yeah,
right.
Yeah.
You know-
That might make
Yeah.
these workshops really unique. You're doing hands-on science and you're learning it through the lens of a biblical worldview. which is
Exactly.
awesome.
Yes.
So it's
People love
something
that.
very unique, and you wouldn't get this very many places. You know, in the
I don't
media
know of
today,
anywhere else.
even at other museums, you know, this is something very unique.
Yes. Yeah.
So
Yeah,
we
thank
have
you,
You
pinned,
did great, Avery.
Thank you.
Very
Good
proud of
job.
you.
Taught, and
Yes,
I
good
really
job.
enjoyed that.
Yeah.
we have
Fantastic.
pinned a link to creationmuseum.org/camp for those who want to learn more about the camp. And then there's also other workshops too that you can learn more about from a day-to-day basis as well. So thank you very much for joining us today.
Yes,
thank you,
thank you.
Jennifer.
Thank
you,
you.
Avery.
My pleasure.